Greetings everyone. This video is for Activity 7, the morphological examination of abnormal RBCs and WBCs in blood smear. Like your Activity 6, you should also draw abnormal RBCs and abnormal WBCs using your worksheets. After you draw the abnormal RBCs and WBCs, you should scan your worksheet and submit it virtually to your instructor. So let's start with our lecture discussion. Poikilocytes. Poikilocyte is a term which means variation in shape. By the way, variation in size is called anisocyte. Okay, so take note of that. Anisocyte is variation in size, and poikilocyte is variation in shape. And we're talking about our RBCs here, not WBCs, but RBCs. So if you find poikilocytes in your peripheral smear, then it presents visual clues for what might be the source of patients' hematologic problems. And sometimes it can be caused by decreased RBC cell destruction, increased destruction, or defective splenic function. So first, poikilocyte that I will be discussing is your acanthocyte. So again, take note that the following RBCs, okay, that I will be discussing in this next slides are poikilocytes. Okay, so number one, we have acanthocyte. So as you can see in the picture, okay, this one is the acanthocyte or spur or thorn cell. So your acanthocyte again can be called spur cell or thorn cell. Okay, it is characterized by having a spheroid shape with 3 to 12 irregular spikes, and it is due to increased cholesterol content inside its RBC membrane. Also, other than that, there is abnormal ratios of membrane lecithins and sphingomyelins in the RBC membrane. Next, we have echinocyte. Echinocyte is also known as Burr cell or crenated RBC. Okay, as you can see, unlike the acanthocyte, the projections are shorter compared to acanthocyte, and the projections, the spikes, are distributed equally. Okay, so it has 10 to 30 scalloped short projections, and they are evenly distributed around the surface of the RBC. The cause of this is due to depletion of ATP, okay, because we all know in our lecture class that RBC do need energy in the form of ATP. And also, echinocytes or crenated RBC can be caused by exposure in hypertonic solution. So if you expose your cells, your red blood cells in hypertonic solution, okay, chances are it will be crenated. Okay. Another cause of having an echinocyte in your peripheral smear is having an artifact or being an artifact due to air drying. Next, poikilocyte is your caudocyte. Caudocyte is otherwise known as target cell or Mexican hat cell. Okay. So, Mexican hat, as you can see in the picture, it's like if you're going to look at a Mexican hat on top view, then it looks like this. Okay, and also, okay, did you see the red one flashing in the cell a while ago? That means, of course, you're targeting it. So target cell, so that you can have an idea on what are or what are the other names of this particular poikilocyte. So again, caudocyte can be called target cell and can also be called as Mexican hat cell. It is characterized by having a peripheral rim of hemoglobin surrounded by clear area and central hemoglobinized area. And as far as you can see a while ago, it's like having a bull's eye, okay, due to the thought of having it targeted, okay, in the middle. And then it can be a bell shape using a scanning electron microscope, and it is caused by increased cholesterol and phospholipid in the RBC membrane. Also, another characteristic of podocyte is that it has an excess surface membrane volume ratio. Next is spherocyte. So sphere, its shape is like a sphere. So it is characterized by having a low surface area to volume ratio, and the cause of this is due to defect of loss of membrane due to spectrin loss. Okay, and then decrease also surface to volume ratio, just the same, low or decreased surface area to volume ratio. Next is stomatocyte, this one. So it is characterized by having a mouth or slit-like pallar area with ball-shaped, and it is due to increased permeability of the membrane to sodium. Next, we have our elliptocyte. It is a rod-like shape or even a cigar shape, okay, narrower than ovalocyte. So although you can see that it's like an oval, but it is narrower compared to the ovalocyte. Okay, so the defect here is considered to be in the cytoskeleton. Okay, decrease in the membrane protein band 4.1. So take note that in the RBC membrane there are a lot of parts there, and when there is decrease in membrane protein band 4.1, which is one of those parts that you can see in the membrane of RBC, then the shape of your RBC will be affected, and it will look like this one, okay, an elliptocyte. Next, ovalocyte. So as you can see here, this is your elliptocyte. For sure, okay, the ovalocyte is wider compared to the elliptocyte. So it's an oval-shaped RBC, okay, wider than elliptocyte, and the cause of this is reduction in membrane cholesterol. Next, we have schistocyte or schistocyte. Okay, it is due to fragmentation produced by damage of RBC by fibrin, altered vessel walls, and prosthetic heart valves. So again, to explain this further, when your RBCs are being trapped in the vessel walls, they tend to also change in shape, okay, because they are going to be fragmented, especially if they are bumped with a lot of walls, vessel walls, and even prosthetic heart valves. If they are bumped to those valves, then chances are your RBCs will have a damage of its surface or its membrane, okay, and it will be fragmented. So those fragmented RBCs, you call them schistocyte. The cryocyte, by its word cry, so you should have an idea that when you see that term, the cryocyte, the other term for that is teardrop shape, teardrop cell. Okay, so again, another term for the cryocyte is teardrop cell. Okay, so it is due to squeezing and fragmentation during splenic passage. So sometimes when our RBCs pass through our spleen, they tend to squeeze through the cells there or the parts of your spleen, and the shape will also be affected and becomes like this one, the chryocyte. Next, we have drepanocyte, or this is the sickle cells or the crescent-shaped cells. Okay, it's of course the shape is crescent-like, and of course it is a very alarming sign if you find these RBCs in the peripheral blood, because the patient might have sickle cell anemia. Okay, so it is due to presence of hemoglobin S inside the RBC, and in the absence of oxygen, hemoglobin S polymerizes into rods, okay, causing sickle cell shape. So when these RBCs, okay, the pranocytes are passing through narrow blood vessels, they will actually be trapped because a drepanocyte cannot be is not flexible enough to squeeze in those narrow blood vessels. So of course, when you have sickle cell, you will always feel the pain when these RBCs, these drepanocytes, will be trapped in one area of your blood vessel. Next, we have keratocyte. Okay, so this one. Okay, so keratocyte is actually part of those that can be called schistocyte. Okay, so keratocyte is also known as bite cell or helmet cell. Okay, keratocytes are cells which have been damaged due to contact with fibrin strand. So if there are a lot of fibrin strand, okay, and the RBC happens to go through that particular area where there is a lot of fibrin strand, then the walls or the membrane of the RBC becomes damaged. And once it is damaged, okay, it becomes a bite cell or a helmet-like cell, or should I say keratocyte. Now we have rouleau formation or true rouleau formation. So true rouleau formation are most of composed of RBCs, okay, in a proper viewing area, and it looks like a stack of coins. Okay, so I think you have an idea already of what is rouleau formation. So can I move forward so that you can look? Okay, this one is a rouleau formation. So those are RBC, okay, that looks like a stack of coins. Okay, so going back. Okay. So next we have autoagglutination. So when the cells are clumping together rather than forming a stack of coin, then it is probably caused by autoagglutination. Okay, so autoagglutination is caused by presence of antibody in the plasma. So whenever our body is actually exposed to any foreign body, our body's mechanism is to make antibodies against that. So if the body has these new antibodies, okay, that is forming inside our blood or in the plasma, okay, one particular reaction or one reaction that can happen is autoagglutination. Okay, so this one is a picture of autoagglutination. So again, the RBCs are clumping together, but they are not forming like a stack of coins. Okay, so here in this picture, you can see a lot of variations. Variation in size, in shape of RBC, and then we also have inclusions. Like sometimes our RBC do have inclusions, but normally it shouldn't have any, right? Okay, it doesn't have any granules, right? So if there are inclusions, then we also have terms for them. So if there are siderotic granules inside your RBC, okay, these siderotic granules are also called Pappenheimer bodies, then of course it is abnormal, okay? So we call these granules as Pappenheimer bodies, okay? And then we also have Cabot ring that looks like this, or sometimes it's an 8 form. And then also we have basophilic stippling. Okay, this one is a basophilic stippling, and then Howell-Jolly body. Okay, so for those RBC that has hemoglobin nopathies, then this one will be the form of your RBC when it has hemoglobin SC, and this one will be the form of your RBC when it has hemoglobin C. Okay, for hemoglobin distribution, of course, okay, the color of our RBC is dependent on the hemoglobin content in our RBC. So less the paler it is, of course, then the hemoglobin is less, okay, in that particular RBC. So these are the gradings for hypochromic. So if you find hypochromic cells, then you have to grade them. So what is the grade of your hypochromic cells? So if you find that this is the color of your RBCs, then you grade it as 4+. If it is like this, then 3+ hypochromia, and then 2+ hyperchromia, and 1+ hyperchromia. Okay, so for the size, of course, for variation in size, these are called anisocytes, no? So this is the normal RBC size. If it is too small compared to the normal, it is called microcytic or microcyte. If it is bigger than the normal, then of course it is called macrocyte. If it is big and at the same time it is like, if it looks like an oval, then you can call it as oval macrocyte. And this one, if the size of your RBC is bigger than normal, okay, and the color is, or it is, if the RBC is considered as hypochromic, then you can call it as hypochromic macrocytic cell or hypochromic macrocyte. So we move on to abnormal WBCs. Okay, so here we have a Pilger-Hewitt cell. It is a WBC, okay? A WBC that has 2 lobes, okay? So normally our neutrophil, particularly neutrophil, it has 3 to 4 lobes, okay? So when the number of lobes reaches 5, we call that hypersegmented neutrophil. But if it is only 1 or 2, it is called hyposegmented neutrophil. Okay, so a Pilger-Hewitt, although it has only 2 lobes, okay, it is somewhat abnormal, but the function of this WBC or this neutrophil is normal. Okay, so it looks like a dumbbell or a peanut-shaped nucleus. Okay, by the way, this is called a pince nez, okay, eyewear. Okay, before around 1900s, they used this one as their eyeglasses. Okay, this is called pince nez. The the nucleus looks like this one, pin's knees. That is what the usual characteristic of the nucleus of Pilger-Hewett. So usually, when a patient has Pilger-Hewett anomaly, it is really, it is often, or these Pilger-Hewett cells are often seen in their blood smears. Okay, so again, although its morphology is abnormal because usually neutrophil do have 3 to 4 lobes, but the function of this Pilger-Hewett is normal. Next, we have, again, hypersegmented neutrophil. When the lobes of, or the nucleus of a neutrophil reaches 5 or more, it is already called hypersegmented neutrophil. Okay, what is the cause of this? It is due to abnormal DNA synthesis, and usually this can be found in patients with megaloblastic anemia. Okay, next we have our rods. Okay, as you can see here, these in the cytoplasm area are our rods. Okay, they're rods, okay, small rods in the cytoplasm of your WBC. So the color of this particular rod is pink or red, and it is again rod-shaped in structure. So what are these actually? It is composed of fused primary granules, and when you find our rods, okay, you can have an idea that the patient might have abnormal monocytes or neutrophil. Okay, those maturing neutrophils and macrocytes that are supposed to be inside the bone marrow, they are immaturely released in the blood stream. So this is usually found in patients with acute microcytic leukemia, but again cannot be found in the lymphocytic leukemia. Okay, only for microcytic leukemia and myelocytic rather, and monocytic leukemia. Next, we have Dowell bodies. So this blue inclusion here, okay, is called Dowell body. If it is only one, Dowell body, but if there are 2 or more, then, or multiple blue inclusions in the cytoplasm area, you call them as Dowell bodies. So what are these? These are aggregates of your free ribosomes of your RER. Okay, and usually these are seen in patients with severe infection. Next, we have basket cells. Okay, so actually the cause of this is the degeneration of your nucleus. Okay, your nucleus has been degenerated, and when it is, then it can be it can look like this one, a basket cell. So sometimes when this is not the case, okay, it is due to pressure in making the smear. So if you can still find basket cell, but you know for sure that the nucleus are not degenerated, then the cause of it is probably pressure in making the smear. So too much pressure in making smear can also cause basket cells. Okay, so setting that aside, okay, when it is really caused by degenerated nucleus, then okay, it is usually seen in chronic lymphocytic leukemia or CLL. Next, we have smudge cells. Okay, the nuclear remnants of lymphoid cell. It resembles a thumbprint appearance, and like basket cell, it can also be caused by pressure in making smear. But if that did not happen, okay, if there's no pressure, okay, too much pressure in making smear, then it could be abnormal, or it could be because of remnants or nuclear remnants of lymphoid cell. And when you have these, okay, pathologically, they are seen in patient with chronic lymphocytic leukemia, or CLL. Okay, so there you have it, the abnormal RBCs and WBCs. So if the pictures or the abnormal RBCs and WBCs that I have discussed are not enough to complete all those circles in your worksheets, you can always refer to your hematology atlas or any hematology book. Okay, you can always look for, for any abnormal WBCs and RBCs in the internet. Okay, so just make sure you label your cells. If you can even label the parts inside the cell, that would be better. Okay, so at least you can have an idea of what are those abnormal cells or blood cells that can be found in the smear. Okay, so that's all for today. So thank you so much for listening, and God bless.